Okay, so now that we went into the anatomy and histology of the nervous system, we're going to talk about the function. So a lot of the ions you're going to see again that we talked about with muscle contraction, sodium, sodium flowing in and potassium coming out of the cell. That stuff is very similar. Now we're going to get, it's kind of nice because we kind of, now we'll learn the why of what we had questions on before. So this is at resting potential. Or you can say this is polar. Polar is anytime you have a slightly negative and slightly positive charge. Okay? This right here would be during depolarization, and then this would be repolarization. So before action potential happens, you have potassium. This is potassium by this structure here. You have potassium inside your neuron, and you have sodium ions outside of your neuron. The reason why it's positive outside and negative inside, even though because both sodium and potassium are both +1, is because you have more sodium outside than you have potassium on the inside. So really what it is just more positive than inside of it, if that makes sense. You just have more positive ions here. So when the impulse starts, so it could be anything as like any current could start by, like, let's say touching something that's hot. So the receptor cells, what would happen is sodium ions would start flowing into the muscle, or I'm sorry, into your nerve fiber. So as sodium goes in, now all those positive charges are inside. So now you have sodium and potassium both inside, and you depolarized your neuron. Depolarized because it was negative, now it's positive. You depolarized it. And now it's very positive inside because not only do you have potassium in there, but now you also have sodium. This is action potential, and this is the energy that's going to swoop through your neuron, and if it was muscle contraction, would go into your muscle fiber. After it goes through, then it would become repolarized. Potassium would be like, whoa, it's way too positive in here, and then there's a pump, and potassium would flow outward. Okay, and then it would become negative and positive again, and then eventually everything would go back to the way it started. So it would be potassium on the inside and sodium on the outside. So this is what it would look like going from neuron to neuron. Essentially, just imagine sodium going in and flowing through, and it would jump from this neuron to the dendrites of the next neuron. It would do that through a neurotransmitter. It would come in here, open it up, sodium would flow in, potassium flow out, and then it would continue in, and then it would go down your axon to your axon terminals, and then it would move to your next neuron, the dendrites. Same kind of effect as what we talked about before. Just another picture. Just make sure, some things to note, make sure you know what ions are inside the cell versus outside. This would be at resting potential or polar. It's positive versus negative. Remember the reason why. There's just more positive ions on the outside. And then you can kind of see sodium coming in and causing that positivity. Okay, so your body electricity, just to kind of explain the backstory of it all. So a human body is electrically neutral. That means you have this, usually in your body you have the same number of positive and negative charges. However, there's some areas as your neuron that has a charge will dominate, and this will cause opposite charges to react. The flow of this electrical current will cause energy. So in this picture here, you can see the sodium ions versus the negative. The potassium ions are here, sodium ions are here, and just seeing this is your membrane, and this is how they move throughout inside the membrane. I give this diagram to you in your notes. What you have here is sodium ions on the outside, potassium on the inside. So this would be at resting potential, and it would be and this would be polarized. So this is polarization here. Once the nerve impulse begins, you would depolarize it because sodium flows in, because potassium is already in here. So with potassium and sodium inside the neuron at the same time, that would quickly make it positive, and that would move throughout your cell. And then behind it, potassium would be like, whoa, it's way too positive, and start flowing outward, and then it would go back to being repolarized. The ions aren't in the same place yet, but eventually everything would move slowly back to where it was. So here it kind of shows you step by step. So neuron membrane is at resting potential. That means it would be polar. Threshold stimulus is received. you touch something that's hot, or you see something that you like, or something happens that stimulates it, a thought. The sodium ions, the sodium channels would open. Sodium would diffuse inward, depolarizing the membrane. So now it's extremely positive on the inside of your neuron. The potassium channels would then open, and then that would cause the potassium to diffuse outward, repolarizing the membrane. This bioelectrical current is action potential, and it'll just travel along the axon. So this, you can kind of see the movement occurring. So you can see this is the sodium flowing in, positive, positive, and that would come in through the dendrites, the soma, down the axon, and then all this is like the electrical current moving through the cell. And then just imagine it again. So this would be sodium flowing in, depolarizing, 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 sodium flowing in. Okay, so there's some things that would influence your nerve impulse for speed. The myelin will do that actually. So sodium flowing in, and then the myelin's almost like a slide. When it goes through, he'll shoot through, and then the nodes of Ranvier, the sodium will flow in again, and then it would shoot through, and the sodium would flow through in. So it kind of just speeds it up. So when as an area of myelin, it would cause that positivity to shoot through the neuron. The two junctions between the neuron that I was showing you guys earlier, it's called the synapse. The nerve pathway, the nerve impulse travels from neuron to neuron. If it was muscle contraction, then it would, at the very end, the effector organ, which if it was muscle, then that would jump into the muscle, causing the sarcoplasmic reticulum to release calcium. But this is actually the pathway, so dendrite to the cell body, along the axon to the synapse, which is that gap, into the dendrite of the next neuron. To complete the signal, so sodium coming in, this is sound familiar, but it's going to get to the synaptic vesicles. There'll be neurotransmitters inside. If it was muscle contraction, there'd be acetylcholine, but there's all kinds of different neurotransmitters, and that would cause the release of the neurotransmitter to attach to receptors and causing sodium to flow in, and that would continue action potential. And again, so this is your synaptic vesicles with your neurotransmitters inside of them. We'll talk about this. We'll do this tomorrow. Okay, so there's different types of neurotransmitters. You have excitatory and inhibitory. So excitatory would mean that it would increase the membrane permeability, so that would cause the flow to go even. So you would have like an increase in action potential. That means it's probably something that make you feel happy, excited. That'd be like your epinephrine and your norepinephrine. And you have inhibitory that would decrease the cell's permeability and the chance for the threshold to be, so that'd be like trying to like slow you down, so like serotonin or things like that. So you can kind of see here, it kind of shows all the steps, which is nice. So you have action potential all the way down, and then when you get to the axon terminal, the neurotransmitter would attach, and depending on if it was inhibitory or excitatory, it would either increase action potential or decrease it. Okay, so the last thing we're going to talk about is the reflex arc. So just imagine action potential starting. This would be like your receptor cells, and that would transfer to your afferent neuron. So sodium going in positive all the way through, and then it would get to your spinal cord. This is your interneuron here, and then this would be your efferent neuron back, and then your effector organ, which would be like your muscle, would contract, right? So this is a rapid, predictable, and automatic response to a stimulus. It will occur over a natural pathway, and this is the reflex arc. So this is anything that happens very quickly. We'll learn later that this happens due to fight or flight. Okay, so you have different types. You have the reflex arc, that's only a few neurons, as I just showed you. You have reflex behavior, that's automatic subconscious responses. That's for fight or flight, so jumping out of the way of a moving vehicle. The knee-jerk reflex, it maintains that you stand upright, and withdrawal reflexes. We'll have a lab for this on Thursday where you'll learn all the different type of reflexes. So you have your, just imagine that this would start the stimuli. So this would start sodium flowing in. touch something that's hot, right? So sodium would be flowing in. This would be your afferent neuron. It would travel all the way to your spinal cord. It would transfer, there'd be a neurotransmitter here. It would acetylcholine. It would transfer from this neuron to the next, to the interneuron, and then another neurotransmitter would attach, and it would continue to the efferent neuron. It would come here, right, to the axon terminal, and just imagine acetylcholine attaching to the receptors. That sodium and potassium would keep flowing into the muscle fiber, causing sarcoplasmic reticulum to release calcium, and that would continue action potential. And just imagine the sarcomeres contracting, and what's going to happen to your hand. It would literally move up, right? It would contract your bicep, causing flexion, and you would move your hand out of the way. This isn't in there, but I just wanted to show you the different type of neurotransmitters: acetylcholine, norepinephrine, and dopamine, and then the serotonin. I was telling you this, so this would be like an inhibitor. Okay, so that's it for now. We'll spend all day tomorrow practicing this, and then you have a lab on Thursday. So we'll see you soon.